The sliding filament mechanism. This will be how we are able to begin to produce our muscle contraction. So this provides the contraction and the relaxation of the skeletal muscle fibers. During muscle contraction, myosin cross bridges will pull on actin filaments, causing them to slide inward towards the H zone. And then the Z disc will come towards each other and the sarcomere shortens. But the myosin and actin filaments do not actually change length. The sliding filaments and shortening of the sarcomeres causes the shortening of the whole entire muscle fiber and ultimately the entire muscle. This is called the sliding filament mechanism. So the progressive overlap of the thick and the thin filaments pulls the A disc towards the center of the sarcomere. Or the Z disc towards the center of the sarcomere. So you see, we're pulling towards the center of the sarcomere here. So relaxed, partially contracted, and then maximally contracted. So the Z disc begin to pull towards the center. And they will result in the length of the fiber and muscle. And then we have a couple of things that we need to have go on for this mechanism to occur. First, we have to have the myosin head hydrolyze ATP or go through ATP hydrolysis. This will provide the energy and the orientation. Next, we need to create an attachment of the myosin head binding to the actin, forming what we call a cross bridge. And so this bound actin and myosin will then perform a power stroke where we'll have the myosin head pivot and pull the thin filament past the thick filament towards the center of the sarcomere. And finally, we have to have detachment of the actin and the myosin from one another, and this happens as the myosin head binds ATP, and so it will then detach it from the actin. Remember that your myosin head had a binding site for both actin and ATP. So this link, as well as a link I'll provide on Canvas, will show you this power stroke or this sliding filament mechanism in action so that you can actually see what's going on. Now, that sliding filament mechanism, that's what actually moves the myofibrils. We now have to have what we call excitation contraction coupling go on to actually help us produce this contraction of the muscle. And so in this, we'll have an increase in our intracellular calcium. This increase in the intracellular calcium is actually what starts the contraction. As calcium increase occurs because we've released it from the sarcoplasmic reticulum, we have active transport pumps restore the calcium levels. And we then have calesquitrin help to concentrate the calcium near the site for release at the cytoplasmic reticulum. And so this allows us to be able to actually begin and start our calcium release. Now, Let's talk about how length and tension of the muscle will determine or help to determine the force of the muscle contraction. So we have this here showing our understretched muscle, the optimal length, and then the overstretched muscle. So we're showing how here we overstretch the thin and the thick filaments, and then the understretch. The forcefulness of a muscle contraction is going to depend on the length of our sarcomeres within the muscle before the contraction begins. So you see here at optimal length, we are able to get the most forceful contraction or have the highest tension developed. Now, the neuromuscular junction. When we spoke about, no, we haven't spoken about nervous tissue yet. Sorry, that's my other class. When we talk about nervous tissue, we'll talk about this action potential and we'll talk about this junction again. But we have to have a nervous signal sent to our muscle, and then we'll produce an action potential. With our muscle action potential, we release calcium ions from the sarcoplasmic reticulum that combine with troponin, causing it to pull on tropomyosin and change its orientation, thus exposing the myosin binding sites on actin and allowing actin and myosin to bind together. The use of calcium ions to remove the contraction inhibitor and join the actin and the myosin constitute what we call the excitation contraction coupling, and that's the step that will connect excitation, so the muscle action potential propagation, through the T tubules to actually contract the muscle fiber. Calcium ion active transport pumps will return calcium ions to the sarcoplasmic reticulum. So, the events that we have to have go on to produce this action potential are going to be the voltage-gated calcium channels in the neuron's synaptic end bulbal open, resulting in an influx of calcium, causing exocytosis of our neurotransmitter acetylcholine into the synaptic cleft. The neurotransmitter acetylcholine will bind to the ligand-gated sodium channels on the motor end plate, which causes an influx of sodium into the muscle. This depolarizes the muscle and will result in calcium release from the sarcoplasmic reticulum. The neurotransmitter acetylcholine will be broken down by acetylcholine esterase. So here we see these steps in a figure. So we're in the actual neuromuscular junction here. So the nerve is coming to the muscle, and then we see the axon terminal and the synaptic end bulb of the nerve, and we see our muscle here. We have the acetylcholine released from the synaptic vesicle into this synaptic cleft. The acetylcholine release binds to its receptor, and we have we've released the sodium into the muscle, and we get our action potential produced. Remember, we break down the acetylcholine to turn this off by acetylcholine esterase. So here we have a summary of what's going on in our muscle fibers. Now, we also need to make sure that we talk about depolarizing phase, repolarizing phase, and the after hyperpolarizing phase. So in each of these phases, this is where we can create the muscle contraction. So depolarization, repolarization, and after hyperpolarizing, those will be for skeletal muscle. But when we talk about cardiac muscle in AMP2, you will also see those there. So you'll want to make sure that you know the components of the action potential. All right, so we've summarized here, and we know that we have to have in this excitation coupling mechanism and in our contraction and relaxation. We have to have the brain and the associate neurons coming to the muscle. We must have the acetylcholine, its receptors and enzymes, and then we have to have sodium and potassium channels opening and then closing to create the depolarization and repolarization. We also need the sarcolemma components of the thick and the thin filaments, and we need the myofibril components with our proteins that will allow for the binding. And all of this can only be powered with ATP. We also have muscle fatigue. So if we are unable to maintain the strength of contraction or tension, then that is called muscle fatigue. And it will occur when muscle has low calcium, creatinine phosphate, oxygen, and other nutrients. So we'll talk about why you might see that muscle fatigue after we talk about how muscles create their metabolism.